Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear students, we are in a continuation of the chapter that is mineral nutrition, and today we are going to talk about the metabolism of nitrogen, which is one of the important part of this chapter. Okay. Now, when we talk about the metabolism of nitrogen, how this nitrogen is going to be metabolized and used in plant, that we have to talk here. Okay. Now, so metabolism of nitrogen, in this if we talk about a nitrogen first, it is an essential mineral element required in the largest quantity by the plant because majority component if you to talk there is a presence of this particular nitrogen. So apart from carbon, hydrogen, oxygen, nitrogen is the most prevalent element in the living organism. That also we know because nitrogen is a constituent. You can see this particular diagrammatic picture that the oxygen 65%, 18% carbon, hydrogen 10%, 3% nitrogen, and others. So nitrogen is also apart from the carbon, hydrogen, oxygen, one of the most prevalent. And nitrogen is a constituent of amino acids, amides, we can say nucleic acid, enzymes, proteins, hormones, chlorophylls, and many vitamins which has a nitrogen as their constituent. So if we talk about the plants, plants compete with the microbes for the limited nitrogen that is available in the soil. Actually, nitrogen is present in the atmosphere. Now from atmosphere it is fixed, then reached to the soil, and from soil it is used by the plants. So in particularly we can say the plant compete with the microbes which are present in the soil or available in the soil for the limited nitrogen, and because of this region it is considered as a nitrogen is a limiting nutrient for both natural and agricultural ecosystems. For both natural and agriculture it is a, we can say, the limiting nutrient. Now this nitrogen, it is considered as exists as a two nitrogen ion, that is a nitrogen exists as a two nitrogen atom that is joined by a very strong triple covalent bond, that is n triple bond on n, and it is present in 78% in the particular atmosphere. So this is the nitrogen constant in the atmosphere, how it is present. Now if we talk about nitrogen cycle, then we have to draw a nitrogen cycle, then it will be easy for us to understand. So first if we talk, there is a atmospheric nitrogen, and this atmospheric N2 if we talk, then obviously it is 78%, that is N triple bond and it is present in the form of this and 78%. Now it is known that the biological nitrogen fixation take place, and through biological nitrogen fixation it is known that this nitrogen is converted into the NH3 or fixed in the form of NH3. Otherwise, apart from biological, it can also happen that through industrial nitrogen fixation it can lead to formation of this NH3. Electrical nitrogen fixation, and this electrical nitrogen fixation form a nitrate. Now this NH3 which is formed here, that get converted into the nitrite, and further it is converted into the nitrate, and this whole process it is known as a nitrification. Okay, so now here it is known that this particular part, if we talk, it is a soil nitrogen pool. So this NH3 can be formed, and from this nitrate can be formed. Now this nitrate uptake take place, and by uptake or absorption it become a part of a plant biomass. So when it become a plant biomass, now it has a two way: either it undergo death and this nitrogen can become a part of decaying biomass, or it become a part of animal biomass. When animal eat this plant, the nitrogen enter in the animal and become a part of animal biomass. Ultimately, animal will also go and death. So by this pathway also nitrogen enters the decaying biomass. From decaying biomass, the next one process happens, that is ammonification. And through ammonification, the nitrogen again converted into the NH3. So this pathway goes on, that from atmosphere nitrogen is taken by biological or this industrial or electrical. Industrial electrical are also known as abiological nitrogen fixation. Then NH3 will form. NH3 get converted into the nitrite, then to the nitrate, then by the use of microbes. Then their uptake happens, plant biomass, animal biomass, or decaying biomass. Then again into the soil that nitrogen will reach. But if this keeps on using, we keep on using the nitrogen from the atmosphere and recycling it in the water, particularly soil, then the one way use of this nitrogen can lead to the depletion in the nitrogen content in the atmosphere. So one more process takes place, that is known as denitrification. And through denitrification, the nitrogen, free nitrogen, is released by the breakdown of the nitrate through this process. So this is the whole nitrogen cycle. Okay, so how this nitrogen cycle looks, that we have discussed, how the nitrogen use, how nitrogen pass on to one, from one stage to another stage, that all thing we have discussed. Now the nitrogen fixation we have just seen in the in the nitrogen cycle, that biological, industrial, and electrical nitrogen fixation takes place. So this nitrogen fixation can be biological or abiological. So if we talk about the first abiological nitrogen fixation, which we are going to discuss, abiological nitrogen fixation. Abiological nitrogen fixation means here living organism are not involved. The living organism are not involved in carrying out this nitrogen fixation. Now this abiological nitrogen fixation, if we talk, it is of two type. It is again of two type. And this two type abiological nitrogen fixation, if we talk, then it can be natural, or we can say electrical, and industrial, or we can say artificial, or industrial. So this two type of abiological nitrogen takes place. One is natural, another one is natural is also known as electrical nitrogen fixation, and industrial is also known as artificial nitrogen fixation. Now in this first we are going to discuss, that is natural or electrical nitrogen fixation. In natural or electrical nitrogen fixation, if we talk here, then dear student, in natural or electrical nitrogen fixation, if we talk, then it is known that in nature, lightning and ultraviolet radiation, lightning and ultraviolet radiation provide enough energy, provide enough energy to convert nitrogen to nitrogen oxides. Okay, so this lightning and ultraviolet radiation as an energy which can provide convert nitrogen to the nitrogen oxides. So if we understand it through the reaction mechanism, then we come to know that if there is a atmospheric nitrogen and this atmospheric nitrogen react with the atmospheric O2, and when it react with atmospheric O2, the energy from UV and lightning is provided, and when UV and from UV and lightning the energy is absorbed, and this when atmospheric nitrogen react with oxygen, it is going to form a nitrogen oxides. And this nitrogen oxides can be nitrogen oxide, that is NO, NO2 dioxide, or N2O can be there. So this is the reaction how the particularly we can say natural or electrical nitrogen fixation takes place. Now when these nitrogen oxides are obtained, it is known that these nitrogen oxides, that is NO, NO2, N2O, if we talk, react with water vapors, and it is going to form HNO3, HNO2, HNO. Okay, so in this they particularly form. So these different acids are formed by the particular of this reaction of nitrogen oxides with the water vapor. Now these acids of nitrogen, these acids of nitrogen enter into soil along with rain and dissociate. And in this dissociation, you will see that the HNO3 get converted into NO3 minus and H plus. HNO get converted into NO minus and H plus, and HNO2 get converted into NO2 minus and H plus. So in these three they get converted, and now this all these nitrate, nitrite and NO minus, this all are uptaken by plant and utilized. Okay, so this is the thing happens during this particular part. Now it is also known that we remember this atmospheric nitrogen, if we talk, they are also provided by, so if we can write here one important point that industrial combustion, forest fire, automobile exhaust, and power generating station also provide this nitrogen oxides, or also they are the source of these nitrogen oxides. In such a way, this particular whole, we can say the electrical or natural nitrogen fixation happens. Now apart from this, the another one is the industrial or artificial nitrogen fixation. And if we talk about this, Okay. So now the second is artificial or industrial nitrogen fixation. And in this artificial or industrial nitrogen fixation, if we talk here, N2 react with 3H2 and form 2NH3 molecule. Now here, this ammonia is further, this ammonia is further incorporated into urea, that is NH3CONH3, sorry, NH2CONH3. Okay, in this urea, this is incorporated, and further this urea used as fertilizer by farmers. Okay, so this is what artificial or industrial nitrogen fixation, and these two are the abiological nitrogen fixation. Now we are going to talk about the biological nitrogen fixation. So let's talk about the biological nitrogen fixation. If we talk about the biological nitrogen fixation, obviously living organisms are involved here, and very few living organisms can utilize the nitrogen in the form of N2, which is available abundantly in the air. Okay, so this N2 form can be absorbed by a certain bacteria only. So only certain prokaryotic species, if we talk, are capable of fixing nitrogen, and they carry out a reduction of nitrogen to the ammonia by living organisms. So when N2 get converted into the NH3, it is known as reduction. And this reduction, when carried out by the living organism, it is known as biological nitrogen fixation. So what is going to happen in biological nitrogen? N2 is going to convert it into NH3. Now, if this is carried out by the bacteria, then this specifically bacteria or living organism it is carried out. Now, this biological nitrogen fixation is also known as diazotrophy. It is also known as diazotrophy. Okay. Now let's talk further. So this bacteria, we will study that they are found in the roots of certain plants, and in this bacteria surrounding to the roots of certain plant, and they carried out this particular nitrogen fixation. How this is carried out? How this N2 get converted into NH3? So here the enzyme nitrogenase is used. Here which enzyme? Nitrogenase is used, which is capable of nitrogen reduction, and it is presently exclusively in prokaryote. It is found only in case of the prokaryote, not in case of the eukaryote. Okay. So here you can see this is the soil where the nitrogen fixation is occurring, and from the atmosphere N2 is coming, nitrogen fixing bacteria are there, which is fixing N2 into NH4 plus, or from the organic material by the ammonifying bacteria also this NH4 will be available, as we have discussed in the nitrogen cycle. Further, nitrifying bacteria carry out a nitrification and nitrate will be formed. This nitrate will be absorbed by the plant and the root, and it will reach to the root and form the different compounds. Same way denitrifying bacteria will release the N2. That thing we have discussed all in case of the what particular nitrogen cycle. Now this way, this microbe carry out nitrogen fixation, and such microbes are called as nitrogen fixers or diazotrophs. Okay, we can call them that diazotrophs because they are converting N2 into NH3 with the help of nitrogenase enzyme. Now this bacteria, if we talk about converting N2 into NH3 with the help of nitrogen enzyme, so the nitrogen fixing bacteria are this nitrogen fixing bacteria may be free living or symbiotic. Okay, so they can fix a nitrogen, convert N2 into NH3 in free living state, or maybe in a symbiotic association with the other organism. So what is biological nitrogen fixation that we have discussed till now? It is a reduction of nitrogen to the ammonia by the living organism. Okay, it is known as biological nitrogen fixation. Now, if we talk here about the free living bacteria which is carrying out a nitrogen fixation, then first how this bacteria carry out nitrogen fixation that we have to discuss. Okay, so here how first the bacteria will carry out this nitrogen fixation. So the bacteria fix nitrogen in three different ways. In three different ways. The first way is free living nitrogen fixing bacteria. They fix nitrogen in free living way only. Free living nitrogen fixing bacteria. They fix N2 in free living state only. And this free living nitrogen fixing bacteria, if we talk, the examples are Azotobacter, then Basernickia, then Rhodospirillum, Bacillus, Clostridium, Rhodopseudomonas. This all are the different genus of the bacteria which are involved in carrying out nitrogen fixation in a free living state. Okay. Now, the some bacteria are that will also carry out a nitrogen fixation in a symbiotic way. So now the second group of bacteria is the symbiotic nitrogen fixing bacteria. Symbiotic nitrogen fixing bacteria. And if you talk about symbiotic nitrogen fixing bacteria, there are several types of symbiotic nitrogen fixing bacteria and nitrogen fixing association are known. Okay. Now, if we talk about example, then most prominent is legume bacteria relationship. So symbiotic nitrogen fixing bacteria, if we talk, then the most prominent example most prominent example is legume bacteria relationship. Is legume bacteria relationship. Okay, and that is the most prominent example legume bacteria relationship, and it is by root nodule formation. Root nodule are the small outgrowths on the root which are their formation is occur, and in that the particular nitrogen fixation is carried out by the symbiotic bacteria. So if you talk about the bacteria and the type of host, So in symbiotic relationship both get benefited. So if we talk about the bacteria and type of host, then bacteria, in this the first bacteria are the rhizobium. And rhizobium, if we talk, that show a symbiotic association with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover bean, etc. Okay, so these all are the leguminous plant, and with this leguminous plant, you will see that rhizobium show a symbiotic association. Okay, to carry out a nitrogen fixation. Same way, another bacteria are the Frankia, and this Frankia shows symbiotic association with roots of Alnus casuarina, and this Alnus casuarina are non-leguminous plant or non-legumes. Okay. Apart from this, if we take some more example, then there is a third variety of rhizobium which is known as azorhizobium. This azorhizobium bacteria shows symbiotic association with the stem nodule of Sesbania. So in Sesbania, there is a formation of stem nodule in which this azorhizobium carry out a nitrogen fixation. And fourth is bradyrhizobium and cynorhizobium. This bradyrhizobium and cynorhizobium, if we talk, they carry out a nitrogen fixation by forming a root nodule of soybean, and within them this nitrogen fixation get carried out. Okay, so this is the what some of the example which particularly carry out this nitrogen fixation in a symbiotic way. Now in this you have to remember that both rhizobium and frankia are important and that are frequently asked in exam. So you should remember that rhizobium, which is a rod shaped bacteria, and frankia, which is filamentous bacteria, they are heterotrophic, free living, form in they are free living forms in soil, but fix atmospheric nitrogen as symbiont. Okay, or in a symbiotic way, they carry out a particularly atmospheric nitrogen fixation. Now, third way of where a bacteria used to fix a nitrogen, that is a loose symbiotic bacteria. And if we talk about an example of a loose symbiotic bacteria, then this is a symbiosis. It is a symbiosis without nodule formation. So we call it as a loose symbiotic bacteria. And the example is azospirillum with maize root. Okay, so azospirillum with maize root carry out this loose symbiotic association and the nitrogen fixation. Now the other first are the bacteria. Now the second group is the cyanobacteria. Cyanobacteria are also known as blue-green algae. And if we talk about the cyanobacteria, they can be free living or symbiotic. And they particularly, specially, they can be free living or symbiotic, but they fix nitrogen inside specialized cells called heterocyst. Okay, in that they particularly carry out this nitrogen fixation. So this blue-green algae also fix nitrogen fixation in different ways. And if we talk about that, then here, if we take an example of free living nitrogen fixing blue-green algae, then here this free living nitrogen fixing blue-green algae fix nitrogen without help of host. And this free living blue-green algae or nitrogen fixing blue-green algae, if we take an example, then their example are nostoc, Anabina, Oscillatoria, Olozera, Cylindrospermum, etc. But in that, it is known that Oscillatoria and Olozera, they are active nitrogen fixers in paddy fields or rice fields. Whereas the Cylindrospermum, it is active nitrogen fixer in sugarcane and maize field. Okay, so this is the how they to carry out a nitrogen fixation in a free living state. Now, apart from this, there are symbiotic nitrogen fixing bacteria as well. And if you talk about that, then symbiotic nitrogen fixing blue-green algae, if we talk, then they fix nitrogen with symbiotic association. And it is known that blue-green algae shows symbiotic association or cyanobacteria make symbiotic association with almost every group of plants in plant kingdom. Okay, so in plant kingdom, they show almost with every group of plant. Now, if we talk about some of the blue-green algae and their hosts, with whom they show a symbiotic association, and in that The first example is Anabena cycade. This Anabena cycade shows symbiotic association with coralloid root of cycas. This coralloid root are the negatively geotropic root of the cycas. Then second is Anabena azole, and this Anabena azole show a symbiotic association with the fronds or compound leaf of Azolla pinnata. So here you should remember that this Anabena cycode shows symbiotic association with the leaves of this Anazolla pinnata, that is a fern plant which is a pteridophyte. Whereas cycade shows symbiotic association with the roots of the cycas. Okay. Apart from this, there are some other example where the third species is Anabena anthocerote, and this Anabena anthocerote shows symbiotic association with the host, that is thallus of anthoceros, and this anthoceros is bryophyte. Whereas fourth, if we talk, that is nostoc species, and this associate with Gunera plant, which is an angiosperm, stem, and trifolium is also angiosperm. With its roots, it shows symbiotic association. So we remember nostoc can show free living as well as symbiotic way they can fix nitrogen. But first three example of symbiotic association you should remember. The last example, this Gunera and trifolium, if you will not remember, then it's okay. But other three examples you should remember here. Okay. So that is what about the particularly free living nitrogen fixing and symbiotic nitrogen fixing bacteria. Now in this, the prominent example is the legume rhizobium relationship, and that we have to understand in a proper way, in a better way. Okay, so that we will discuss in detail. So let's talk about the symbiotic nitrogen fixation, symbiotic biological nitrogen fixation, which we are going to study here. And in that, the most prominent symbiotic nitrogen fixation where we have discussed is the legume bacteria relationship, where a rhizobium bacteria shows symbiotic association with the roots of leguminous plant. And in that, you will see that the legume based cropping system is there, in which you will see that this is the particularly nodules, okay, in which the biological nitrogen fixation takes place and N2 get converted into NH3. Either through decomposition, it will add nitrogen to the soil and it will be used by the plants, or after particularly passing to the different plant organs, the death of them it can occur, and from the crop residue also it can add a nitrogen to the soil, which is used by the particular different crops. So here, species of rod-shaped rhizobium has such a relationship with the roots of several leguminous plants such as alfalfa, sweet clover, as we have discussed, sweet pea, lentil, garden pea, broad bean, clover bean, etc. This is alfalfa, this is sweet clover, this is sweet pea, okay. This is garden pea. You should see the diagram properly, okay. Then this is the garden pea, okay. Then this is the beans, this is the lentils. With all them, this particularly rhizobium can show symbiotic association. So most common association on root is as a nodule. They carry out a nodule formation. Okay, these are the, you can see, these are the nodules on the legumes root. See, this is nodule and this is a root. Okay. Now, these nodules are small outgrowth on the root, obviously, and they carry out this nitrogen fixation. So the microspranchia is also there, as we have discussed. This also produces the nitrogen fixing nodules on the roots of non-leguminous plant like Elna schizurina and myrica, which are the particularly different plants. And this spranchia is a filamentous bacteria. Okay, so here you can see this is nodule formation. So both rhizobium and branchia are free living in soil, but as a symbiote they fix nitrogen, as we have discussed. Now, if you cut a nodule, you will notice that a central portion is red or pink. Okay, if we take a cross section, you will see that that central portion is red or pink. Okay, like this. This is the nodule you can see. Okay, and in this you can see this is the red or particularly pink portion. So this is due to the presence of leguminous hemoglobin, which is also known as leg hemoglobin. Now, if leg hemoglobin is there, what is the function of that? See, in nodule, the bacteria are there, and this bacteria is going to fix a particular nitrogen by the use of a nitrogenase enzyme. Now, nitrogenase enzyme is an oxygen sensitive enzyme. It will oxidize, it will not occur in the presence of oxygen. So, to maintain the anaerobic condition, this leg hemoglobin will work, and it will act as an oxygen scavenger. So, this leg hemoglobin act as an oxygen scavenger. Leg hemoglobin, it act as a oxygen scavenger, means it's prevent a particular oxygen to reach to the nitrogenase 1. Okay, so it protect nitrogenase enzyme, it protect nitrogenase enzyme from oxygen by maintaining anaerobic condition around nitrogen enzyme, because it itself reacts with oxygen and does not allow oxygen to reach to this particularly uh, nitrogenase enzyme. Okay, this way it will work. Now, later on, if you talk, now we will talk about the nodule formation. See, in nodule, the nitrogen fixation is going to take place. In nodule, there is a particular this pink or red color pigment that is a like hemoglobin. So, how nodule formation takes place, that we have to understand first. Then we will understand the nitrogen fixation, how it takes place. So let's talk about the how the nodule formation occur in leguminous plant. So nodule formation involves a sequence of multiple interaction between the rhizobium and the roots of the host plant, and host plant here are nothing but a leguminous plant, obviously. Okay. So what are the interaction occurrence that we have to understand? And this interaction are correctly shown in this diagram. See, this is the soil. Okay. This is the particularly soil part. So, sorry, this is the particularly we can say the root. Okay. Root hair, and this is a particularly root part. Okay. So this root hairs, which are the unicellular structure and extension of the epidermal cells. This is the soil particles, and in soil particles you will see the bacteria. Now what happens? This bacteria first what happens? This particularly the curling. So first what happens? This rhizobium bacteria which are present in the soil, they get multiply, okay, and colonize the surrounding of the root and get attached to the epidermal and root hair cells. They attach to this root particularly epidermal cells. Then they further the curling of this root hair will happen, and they invade this particular root hair, and from root hair they enter within the particular body of this particular the particular root. Then they will enter the particular this bacteria enter the root, and this chain of bacteria will be known as infection thread, which contain the bacteria. And this bacteria through infection thread reach to the inner cortex and perimycycle cells. And this inner cortex and perimycycle cells divide, undergo division, and form a mature nodule. Okay, so penetration of bacteria within the root through the root hair and reaching to the cortex and the pericycle cell, and then causing their division and forming a nodule formation. That is the sequence of nodule formation. So this development of nodule in soybean. Now the principal stages in the nodule formation are summarized as below. First, what will happen? Rhizobia attracted by the sugar and amino acid released by the host legume. Means legumes are there, the leguminous root, the legume plant root are there. The root will release some of the sugar and amino acid. So due to the release of this sugar and amino acid, this bacteria, rhizobium bacteria, get attracted towards the root. Okay, and the colonize surrounding the root and get attached to the epidermal and root hair cells. So the root hair curl and the bacteria invade the root hair after the particularly attraction. So we remember this, the bacteria are attracted toward this. So this attraction is chemotactic. Is chemotactic. Okay. Now rhizome, this sugar and amino acid obviously released by the host root. Okay. But in opposite, to identify host root, there is a ricardapsin protein of bacterial cell, that ricardapsin protein of bacterial cell identify host root. Okay, so that is the thing. Then after identification of the host root, the bacteria, the root hair will curl and the bacteria will invade the root hair, means this is the infection. After infection, the infection thread will form, and the infection thread is produced which carrying the bacteria into the cortex of the root. Okay, because how the cortex it will reach, crossing the epidermis, they will enter the cortex, and after reaching the cortex of the root, the bacteria are released from the thread into the cells, and here the bacteria are modified into rod-shaped bacteria which cause inner cortical and pericycle cells to divide. So then the bacteria are released from the thread into the cortical cells of the host stimulus, and the host cells are divided. Okay. So the important point is that we remember the curling here of root hair, curling of root hair is induced by specific complex polysaccharide found on the surface of found on the surface of rhizobia, and these are recognized by lectins, and these lectins are the small proteins of host plant root. Okay, now after the curling, okay, the bacteria will induce a division of a cortical and pericycle cell. So the dedifferentiation of cortical and pericycle cell PGRs, that is plant growth regulators like auxin and cytokinin are required. Now we remember in dedifferentiation, what will happen? The non-dividing cells will be dividing. Okay, and here this particular division occur. Here now we remember that auxin is provided by host root. Auxin is provided by host root, while cytokinin by bacteria. Okay, so cytokinin will be provided by bacteria and the auxin will be provided by the host root, and the division of cortex and the pericycle cell will occur and the nodule formation will take place. Now this mature nodule which is formed is in direct vascular connection with the host for the exchange of nutrient. So be remember, mature nodule has direct vascular connection with the host for exchange of nutrients. Okay, and in that, if we talk, ultimately the nodule formation taken, and in nodule each cell has a particular nitrogenase enzyme. That nitrogenase enzyme has a capacity of the nitrogen fixation. So now the particularly, we as we have discussed, this is the attachment of rhizobium bacteria to here, then the curling of it will occur, then they will enter the root hair and infection thread will be formed, and through action infection thread the nodule will be developed and reify the infection thread. Now here, thus leads to the differentiation of the specialized nitrogen fixing cells, and ultimately the nodule thus formed establish direct vascular connection. As you can see, this is the infection thread and which having a bacteria. Okay. Now the nodule contain all the necessary biochemicals components such as enzyme nitrogenase, leg hemoglobin, which are required for the nitrogen fixation. Now nitrogenase is the important, and from here we can understand the mechanism of biological nitrogen fixation. Now if we talk about the nitrogenase enzyme is the molybdenum ion protein, and this catalyzes the conversion of atmospheric nitrogen to ammonia. So N2 is converted into the NH3 by this molybdenum ion protein, that is nitrogenase enzyme. And this is the structure, detailed structure or protein structure of this particular one. The first stable product of the nitrogen fixation is nothing but a ammonia, which is formed from the N2 through its reduction. Now, it is interesting to note that these microbes live as aerobe. Okay, it is interesting to note that the microbes live as aerobe under a free living condition. Means the rhizobium live is a free living condition. That is a what? Aerobe, where nitrogen is not operation. But during the nitrogen fixing event, when they are carrying out a nitrogen fixation converting N2 into NH3, they adapt to a anaerobic. They adapt to the anaerobic condition, that is bacteria, thus protecting the nitrogenase enzyme, and nitrogenase enzyme carry out a nitrogen fixation. Okay, so during nitrogen fixation, the anaerobic condition should be there. So now, how they particularly carry out nitrogen fixation in anaerobic state, that we have to understand. So this is the example of how the nitrogenase enzyme work. Okay, dear students, be remember, this is the enzyme, that is nitrogenase. Okay, now this nitrogenase enzyme, which is a molybdenum ion protein that react with the substrate, that is a nitrogen gas, that is N2, that is N triple bond N. When N triple bond reacts with the enzyme, it is form a enzyme substrate complex. Okay, this is enzyme and this is a substrate, that is N2. Then reduction occur and 2H plus are added here, which react with 1H plus react with 1 at nitrogen atom. So here 2 nitrogen atom react with the 2 particular hydrogen, and here N2H2 molecule will be formed. Here this is enzyme. Again reduction occur, 2H plus are added, and enzyme will react with enzymes 2 and N2H2 react with 2H plus, and here 2NH2 will be formed. Then here also again the reduction occur and 2H plus are added, and here these are forming a 2NH3. At last the free nitrogenase will be released like this, and the 2NH3 molecule are released here, that is the ultimately we can say the ammonia product. Now this enzyme nitrogenase become free, and they can bind another molecule of nitrogen and carry out a nitrogen fixation, and such a way the nitrogen fixation take place. Now we remember the enzyme nitrogenase is highly sensitive to the molecular oxygen. So it is sensitive to the molecular oxygen. So it requires anaerobic condition. And the reaction, the for the anaerobic condition, leg hemoglobin will work. We have discussed that protect it from the. So when this carry out whole reaction, ultimately the reaction will be like this: that N2 plus 8H plus electron plus 16 ATP give rise to the 2NH3 H2 and 16 ADP and 16 inorganic phosphate. So 16 ATP are used. So per molecule of NH3, per molecule of NH3, 8 ATP are going to be used. That is so because 2 NH3 are produced. So for 1 NH3 synthesis, 8 ATP are used. Okay, so this is how the nitrogen fixation take place. To protect the enzyme, the nodule also contain oxygen scavenger called a leg hemoglobin. Okay, so it protect the enzyme from its particular oxidation. Now this leg hemoglobin, if we talk, that is oxygen scavenger, it is a hollow protein, and this is synthesized by the joint product of both plant and bacterium, in which globin is produced by plant, is produced by host plant, that is legume, and heme, that is an iron atom found in a porphyrin ring, that is produced by rhizobium. So heme is provided by the rhizobium and leg hemoglobin is provided, and hemoglobin is provided by the host plant, and by combination of this the joint product will be formed. So it is also a particularly formed in a symbiotic way, or it is also a product of symbiosis, we can say. Okay, and this way the oxygen scavenger will work. Now we remember during the previous reaction the H plus are getting added for the reduction, that H plus are particularly provided by the NADH, okay, which is produced by the respiration of the host cell. Now ammonia when synthesized, the nitrogenase enzyme that is synthesized by the ammonia required a high input of energy, as we have discussed the 8 ATP for each NH3 produced, and the energy required is obtained from the respiration of the host cell. Okay, so that is the other thing. So this is how the nitrogen fixation take place in this. Now here we have to remember some of the genes. Okay, and if you talk about the genes, we know that it is a segment of DNA which is present, which is coding for a function. And here the different genes are there in this. It is known that node gene is present in both plant and bacteria. Present in both plant and bacteria, that is rhizobium. And this node gene code for the node factor which is responsible for the curling of the root hair. Apart from this, the NIF gene, that is a nitrogenase inducing factor, and fix gene, they are present only in bacteria. Present only in bacteria. Okay, this way the particular nitrogen fixation takes place, and ultimately NH3 gets synthesized by the symbiotic nitrogen fixation. Now after this mechanism of biological nitrogen fixation, now what will happen to this ammonia which is synthesized by the reduction of N2 to NH3 in the diazotrophy, and that we will understand or study in the name of the topic, that is the fate of ammonia. Fate of ammonia will decide what will happen to this particularly ammonia. So at physiological pH approximately near about 7, the ammonia get protonated to form a NH4 plus ion, that is the ammonium ion. So here you can see this NH3 getting protonated and forming the NH4 plus, that is ammonium ion. Now ammonium ion is quite toxic. No doubt, the most of plant can assimilate nitrate as well as ammonium ion, but ammonium ion is quite toxic to the plant, so they cannot accumulate in them. And how this NH plus 4 plus or ammonium ion is used to synthesize the amino acid in plant, that we have to study. So let's see how that particularly involved in a amino acid synthesis, this NH4 plus. So there are two main ways or two main reactions by which this ammonium ion get incorporated into the amino acid or in the amino acid synthesis. And in that, the first reaction is the reductive amination. In reductive amination, the ammonium ion directly react with the alpha ketoglutaric acid and form a glutamic acid. And you can see this is the reaction. Now, what is happening in this reaction? Let's understand. This is the alpha ketoglutaric acid is a 5 carbon intermediate compound, and it is ammonium ion. It is NADPH will be used in the presence of glutamate dehydrogenate, glutamate or glutamic acid amino acid will form along with water is released and NADP is getting released. Okay, so this is the first reaction where the ammonia which is synthesized by the biological nitrogen fixation get converted into the formation of the amino acid, that is a glutamic acid. So glutamic acid is the first amino acid which is getting synthesized. Now from glutamic acid, another all different amino acid, remaining 19 amino acid can be synthesized by the other reaction, that is a transamination. And transamination involves the transfer of an amino group from the amino acid to the keto acid. What will happen? Amino acid which is synthesized, that is glutamic acid, has amino group. So that is transferred to the keto group of the keto acid. And you can see this is the reaction where you can see this is the amino acid, okay, which is amino group, COOH group. And this is the keto acid which is COO group. Now this amino group get transferred to this keto acid. So what will happen? This amino group will be released and it become converted into keto acid, but this keto group will be removed and here amino group will be added. So this become a now amino acid. So this was the glutamic acid and another amino acid will be formed by the reacting with the keto acid. Okay. Now glutamic acid is the main amino acid from which the transfer of amino group take place and other amino acid are formed through the transamination. So if we talk about the glutamic acid and the keto acid, the another amino acid will be formed by the release of amino group, and this glutamic will convert it into the what? Alpha ketoglutarate. And to carry out this transfer of amino group to the keto acid, the enzyme transaminase catalyzes all such reaction. And if you take an example of this one, then you can see here this is the reaction how that is happening. Now, and in this example, we will write to reaction. Suppose we are taking an amino acid that is a glutamic acid. Now, when glutamic acid react with oxaloacetic acid, it is going to form a alpha ketoglutaric acid, and the another amino acid which is going to synthesize here, that is aspartic acid. Apart from this, the glutamic acid react with a pyruvic acid. When it reacts with pyruvic acid, it is going to release alpha ketoglutaric acid and alanine will be formed. Such a way, the alanine, aspartic acid, and other amino acid will be synthesized by this transamination reaction, and they here the particular transaminase enzymes are going to be used. Now, so reductive amination and transamination will form a different amino acid, but it is known that compared to amino acid, amides are better for the transport of nitrogen. So amides are formed. So the first is the what are the amides? So amides are formed by catalytic amidation reaction. Now we have to understand that what will happen to this catalytic amidation. Okay. So amides are also considered as double aminated keto acid. Amides are double aminated keto acid, means they have a two amino group. Now how they are formed? So in this we know that amino acid have a one amino group and one carboxylic group. So suppose this is the what particularly amino acid. Now what will happen? Suppose this is as a OH. If we remove this OH and add another NH3 plus, then what will happen? This will have NH3 plus, CO, NH3 plus, and here the R1. So this is amide. So how the amide is getting formed? The hydroxyl part of amino acid is replaced by amino group. So now the amino acid will be modified amino acid which have a 2 NH2 group or amino group, and that particular modified amino acid with the 2 amino group will be considered as a amide. Because of this we call it as a double aminated keto acid. Now it is known that the two most important amides are the asparagine and the glutamine that are found in a plant, are a structural part of protein. Now this is asparagine and this is a glutamine. You can see here it has a 2 amino group, that is this is NH3 and this is NH2. Okay, 2 amino group are there. Okay, so this 2 amino group, due to this they are known as a particularly what amides. Now these are formed from the 2 amino acid, that is aspartic acid and the glutamic acid. Aspartic acid will form in asparagine and glutamic acid will form a glutamine. Okay, so that are formed in addition to the another amino group to it, and you can see here that this is having a what NH2 group and this is NH2 group, that is asparagine. Okay, that particularly converted into aspartate. Okay, when the amino group is released, and same way glutamate is there, that is converted into the glutamine. Now here the hydroxyl part of the acid is replaced by another H2 radical. Because of this, it is known as a what? We can say the amides. Amides, if we talk, they contain a more nitrogen than the amino acid. What is the important point for amide formation? That amides contain a more nitrogen than the amino acid. So they are transported to the other parts of the plant via xylem vessel. Okay, so they have more nitrogen. Because of this, there is a need of formation of the amides for the better transport. Now, apart from this, there is another component in addition to the amides, that is the urates. So urates, in addition, along with the transpiration stream, the nodules of some plants nodules are there which export the fixed nitrate as a urates. And these urates are nothing but it is a degraded urea, and these compounds also have particularly high nitrogen to carbon ratio. Because of this, they are also better for the transport of nitrogen. So nodules of soybean plant will release such type of fixed nitrogen as a urates. Okay, now after this nitrogen fixation biological NH3 will be formed. NH3 is also formed by the ammonification process. And if we talk about ammonification, it is the process, as we have discussed in the nitrogen cycle, where decaying biomass undergo decomposition and will form ammonia. So decomposition of organic nitrogen of a dead plant and animals that is present in the decaying biomass, it is known as ammonification. So decomposition of decaying biomass and release of ammonia is the ammonification. Okay, so this is the how the ammonification will be. Soil organic matter is there, manure is there, rooting plant residues are there, that will release ammonia. Okay, that will be converted into the ammonium. Okay, so ammonification is carried out by the heterotrophic bacteria. Okay, now in this ammonification, what will happen and which bacteria is carried out this particular process? So it is known that it is carried out by Bacillus ramosus, Bacillus vulgaris, and Bacillus mycoides. So these three are the Bacillus bacteria which are particularly involved in what ammonification. So these all are known as ammonifying bacteria. And in this, what happens? They have a detritus. Now we know that detritus have a protein. So first, proteases will work and they will break the protein into amino acid. Further, this amino acid get broken into NH3 and organic acids. Further, this NH3 get converted into NH4 plus ions. Okay, so this way the breakdown of this detritus and release of ammonia will occur. Now in this, we remember that the breakdown can be both anaerobic and anaerobic. Deammonification can be aerobic and anaerobic. So in this, the anaerobic breakdown of protein is known as putrefaction. So next is what is putrefaction. So we remember putrefaction is anaerobic breakdown of protein. And apart from this, there is a one more term, that's a decay. Decay is an aerobic breakdown of protein. Okay, so this breakdown can be aerobic or anaerobic. Now the ammonia which is formed by the particularly this ammonification, some of the ammonia volatilizes and re-enters the atmosphere. So we remember some of the ammonia volatilizes and re-enters the atmosphere, but most of it is converted into nitrates by soil bacteria, that is nitrification. Means some will enter the atmosphere and some ammonia will undergo the nitrification. And the ammonia which is produced by the biological nitrogen fixation, or we can say the electrical nitrogen fixation, that is also undergo what a particular nitrification. So now the next process which we are going to discuss, that is nitrification. And in nitrification, what happens? The some of the ammonia which is volatilizes and re-enters the atmosphere, but the another remaining ammonia from the ammonification and biological nitrogen fixation, that is converted into the nitrate by a soil bacteria, and that is known as nitrification. Now in this nitrification, two reactions will occur. That is NH3 will react with 3O2 and release a 2NO2 minus 2H plus and 2H2. That is the first reaction, and here this nitrite formation will take place. So from ammonia, first reaction the nitrate will form. This nitrate will react with O2 and undergo a formation of nitrate. And along with it release some energy as well. So these are the chemoautotropic bacteria. And for the first reaction, conversion of NH3 into the nitrite, you will see that there is an involvement of a particular bacteria. So here, the bacteria which are going to be used, that is the nitrosomonas, in this particular first reaction conversion into the nitrate, along with this nitrococcus can also carry out this reaction. Apart from this, you will see that this conversion of nitrite to the nitrate is carried out by the nitrobacter or nitrocystis. Okay, and ultimately the nitrate is going to be formed. So the ammonia is first oxidized to the nitrite by the bacteria nitrosomonas or nitrococcus. These are nitrosomonas, these are nitrococcus, that is a rod-shaped bacteria. And the nitrite is further oxidized to the nitrate by the nitrobacter, okay, or nitrocystis. These are nitrobacter bacteria. These steps are called nitrification, and these nitrifying bacteria which are involved in this, that all bacteria are known as chemoautotrophs. They particularly oxidation of this inorganic compound happens and the energy is released. Because of this, they are known as chemoautotrophs. Now further, the nitrate which is ultimately formed by the nitrification is thus absorbed by the plant and it is transported to the leaves. Okay, and when it is transported to the leaves, okay, that nitrate in leaves is reduced to form ammonia, and that finally forms the amino group of amino acid. Okay, so how this nitrification nitrate will form and how nitrate will be assimilated into the particular formation of the particular amino acid in the plant. So nitrate is the most absorbable form. We know because of this nitrification will produce a nitrate. Then absorbed by the plant, and finally in plant it is further reduced into the amino acid. So now how the nitrate assimilation will occur, that we have to understand. And in nitrate assimilation, you will see that it is a reductive process. And in this, okay, so this is the reductive process. Okay, and what happens in this during nitrate assimilation? First, this nitrate is converted to again nitrite by the enzyme that is nitrate reductase, and this will take place in the cytoplasm of any cell of the plant where the nitrate is available. Okay, after this nitrite is converted into the NH4 plus ion, that is ammonium ion, by the nitrite reductase, and it takes place in the plastid in the leaves. Okay, so nitrite is converted into nitrate, and nitrite is converted to ammonium, and now this ammonium will be incorporated into the amino acid. Okay, so that is nitrate assimilation takes place in the plant biomass. Now from plant biomass, an animal to the decaying biomass, and from decaying biomass how the ammonification will happen, that we have already discussed. Now this is the how nitrogen cycle will take place, and continuously the use of the nitrogen will occur from the atmosphere. Now we have to replenish it also. So the last process which we are going to discuss, that is the nitrification. That is denitrification, sorry. And in denitrification, what will happen? The nitrate which is formed by the nitrification, which is present in the soil, it is also reduced to the nitrogen by a process of denitrification. So this reduction to the nitrogen will take place, and this denitrification is carried out by some of the bacteria, and this bacteria is known as Pseudomonas and Thiobacillus. These both are known as Pseudomonas denitrificans specifically, and Thiobacillus denitrificans. And when they carry out this denitrification, you will see that we have, suppose, NO3 minus, 2 NO3 minus, then what will happen? They will release one oxygen atom. So this will convert it into 2 NO2 if O2 is released. Then one more, okay, O2 if it get released, then they become a 2NO. Then the half O2 will be released, and that converted into the N2. And this N2 reaches to the atmosphere, and such a way the particularly this whole denitrification process takes place. So that is all about the what, how nitrogen fixation carried out, what different type of bacteria are involved, what is ammonification, what is nitrification, what is this fate of ammonia, that all thing we have discussed in this particular part. So last, there are simple some question. Nitrogen exists in two nitrogen atom joined by the triple covalent bond. So triple bond will be the correct answer. Conversion of molecular nitrogen to the ammonia is obviously, we can say, the nitrogen fixation. And ammonia is fixed first oxidized to nitrite by obviously nitrosomonas or nitrobacter. Okay, molybdenum iron protein is nothing but a nitrogenase enzyme which carry out nitrogen fixation, and that is all about the what, a nitrogen metabolism. Okay, I hope you have understood, dear students. That is all about the nitrogen metabolism, nitrogen cycle, and nitrogen fixation, and the fate of ammonia. Thank you.